It's a really sobering thought, isn't it? Cancer. Right up there as the second leading cause of death in the U.S., just behind heart disease.
It is. And the numbers, while stark, don't even capture the full picture. The human costs, the suffering involved.
Exactly. It affects so many people, directly or indirectly, and it always leaves you wondering, when are we going to truly get a handle on these diseases?
That's the driving question, absolutely. And it's vital right off the bat to understand that cancer isn't just one thing.
Right.
Think about Hodgkin lymphoma, often very treatable, even curable. Then contrast that with, say, pancreatic adenocarcinoma.
Worlds apart.
Completely different prognosis, generally much more challenging. That spectrum just highlights the sheer complexity we're dealing with.
Absolutely. So this deep dive, we're not just scratching the surface. We're really aiming to get into the weeds of neoplasia. That's the term, right? New growth.
That's the one, neoplasia.
Our mission today is to unpack the fundamental causes, the actual molecular gears turning behind the scenes. But there's also good news in this story, isn't there?
There really is. Despite the challenges, if you look at the trends over the last few decades, cancer mortality rates in the U.S. have actually been declining for both men and women.
That's encouraging.
It shows real progress, better understanding, much better detection, definitely improved treatments. It's a testament to the research, but, still a long road ahead.
Okay, so to tackle this huge topic, where do we even begin? How do we structure this?
Let's start with the language, nomenclature. Getting the terms right so we're all on the same page.
Makes sense.
We need to define neoplasms, these new growths. And crucially, the difference between benign, meaning generally non-cancerous, and malignant, which is cancerous.
Though even benign can be dangerous sometimes, right?
Yeah.
Like in the brain.
Exactly. Location matters hugely. A benign tumor pressing on a vital structure can be life-threatening.
Okay.
Then we'll look at specific terms. You often see OMA for benign tumors like adenoma for glandular tissue, papilloma for those finger-like growths, cystadenoma if it's cystic and glandular, polyp if it projects from a surface like the colon lining.
Got it. And for the malignant ones?
Generally, sarcoma indicates a cancer from connective tissue, bone, muscle, fat. Carcinoma is for epithelial cancer, skin, lining of organs. Leukemia for blood cancers. Lymphoma for the lymphatic system.
Okay, sarcoma, carcinoma, leukemia, lymphoma.
And then there are some more complex ones. Mixed tumors, like a pleomorphic adenoma in the salivary gland, have different cell types. And teratomas, those are fascinating. They can have tissues from all three embryonic germ layers. Hair, teeth, bone, all in one tumor.
Wow. Okay, so that's the vocabulary foundation. What's next?
Next, we dig into the characteristics. What actually distinguishes benign from malignant beyond just the name?
How they behave.
Precisely. Differentiation is key. How much do the tumor cells look and act like the normal cells they came from? Malignant tumors are often poorly differentiated or even anaplastic, meaning they've lost that resemblance.
They look weird.
Very abnormal, yes. We look for pleomorphism variations in cell size and shape and other signs of anaplasia. Then there's local invasion. Malignant tumors invade surrounding tissues. Benign tumors usually don't. They tend to push things aside.
And metastasis.
Metastasis, the big one. That's the spread to distant sites. It's the defining feature of most malignant cancers and what makes them so dangerous.
Right.
We also look at growth patterns, how well-defined the tumor borders are. Benign tumors are often encapsulated. Malignant ones tend to have infiltrative borders. And functional changes, sometimes tumors produce hormones or other substances.
What about carcinoma in situ? I've heard that term.
Good point. Carcinoma in situ is a critical concept. It means you have cells that look malignant under the microscope, but they haven't broken through the basement membrane yet. They're still confined to the original epithelial layer.
So pre-invasive cancer, essentially.
Exactly. It has the potential to become invasive, but hasn't yet. It highlights that transition point.
Okay, understanding the terminology and characteristics is crucial. Where does epidemiology fit in? The bigger picture.
Right. Epidemiology looks at cancer patterns in populations. Global impact first, how many deaths worldwide each year. And those numbers are projected to increase, unfortunately.
Why is that? Aging populations? Lifestyle factors?
Both, primarily. Aging populations in many countries, plus the continuing impact of factors like smoking, diet, obesity, and others. Then, focusing on the U.S., we look at the most common types.
Which are?
For men, typically prostate, lung, and colorectal cancer. For women, it's breast, lung, and colorectal. Lung cancer is a major killer for both sexes, largely due to smoking, though rates are thankfully decreasing.
And you mentioned mortality rates are declining overall.
Yes, for many common cancers, we see declines in lung cancer mortality, partly due to less smoking, but also better treatments. Colorectal cancer rates are down thanks to screening like colonoscopies removing polyps before they become cancerous.
Screening is key there.
Hugely important. Breast cancer mortality is also down due to screening like mammography and improved therapies. Same for prostate cancer, better detection and treatment options. And cervical cancer, thanks to pap smears and now HPV vaccination.
So progress is being made on multiple fronts. What about the underlying risk factors?
Yeah.
Beyond just age.
The contributing factors. It's a mix of environmental and host factors. Environmentally, we have infectious agents. We'll talk specific viruses and bacteria later.
Okay.
Then the big lifestyle ones. Smoking, obviously. Alcohol consumption. Diet plays a role. Exposure to environmental carcinogens, certain chemicals, pollutants, radiation.
And host factors, things about the person.
Exactly. Age is the biggest single risk factor overall.
Yeah.
Race and ethnicity can influence risk for certain cancers, often due to complex interactions of genetics, environment, and socioeconomic factors.
Yeah.
And then acquired predisposing conditions.
Like what?
Chronic inflammation is a big one. Conditions like chronic inflammatory bowel disease increase colorectal cancer risk. Certain precursor lesions like those colon polyps we mentioned, or specific types of moles for melanoma. And immunodeficiency, a weakened immune system can't fight off cancerous cells as effectively.
Makes sense. And childhood cancers, are they different?
They often are, yes. While tragic, they're relatively rare compared to adult cancers and often have different underlying causes, sometimes involving developmental processes rather than accumulated environmental damage.
Okay, so we have the definitions, the behaviors, the population patterns, the risk factors. Now, let's get to the core. What's actually happening inside the cell? The molecular basis.
This is where it gets really fascinating. At its heart, cancer is a disease of the genome. It arises from accumulating damaged genetic mutations and epigenetic alterations.
Genetic and epigenetic, so changes to the DNA sequence itself and changes to how genes are controlled.
Precisely. These alterations disrupt normal cell behavior. We talk about the hallmarks of cancer, a set of acquired capabilities that allow cells to become cancerous.
Hallmarks, like milestones on the road to cancer.
Kind of, yeah. And these are enabled by two key characteristics. Genomic instability, which means the cells acquire mutations faster.
Fueling the fire.
Exactly. And cancer-promoting inflammation, which creates a supportive neighborhood or microenvironment for the tumor.
Okay. And within all those mutations, are some more important than others?
Definitely. We distinguish between driver mutations, the ones that actually confer a growth advantage and push the cell towards cancer, and passenger mutations, which are just along for the ride they're present, but don't directly contribute to the cancer phenotype.
So you need the right driver mutations.
Yes. And it often starts with an initiating mutation, the first key hit. But cancer rarely arises from just one mutation. It's usually a stepwise accumulation over years or even decades.
The gradual process.
Very gradual. And we also need to consider cancer stem cells, a small population within the tumor that can self-renew and drive tumor growth and recurrence. They're often more resistant to therapy.
Okay, so these driver mutations, what kinds of genes do they typically hit?
They tend to cluster in 4 main classes of genes that regulate really fundamental cell processes.
4 classes, what are they?
First, proto-oncogenes. These normally promote cell growth in a controlled way.
Like the gas pedal.
Good analogy. Second, tumor suppressor genes. These normally put the brakes on cell growth or trigger cell death if something's wrong.
The brakes.
Third, genes that regulate apoptosis, or programmed cell death. Cancer cells need to evade this self-destruct mechanism. And fourth, DNA repair genes. If these are faulty, mutations accumulate much faster.
So gas pedal stuck on, brakes fail, self-destruct fails, and the repair shop is closed.
That's a pretty good summary, actually. And the mutations work differently. Proto-oncogenes become oncogenes through gain-of-function mutations. One mutated copy can be enough to drive things forward.
One hit is enough.
Often, yes. But for tumor suppressor genes, you usually need loss-of-function mutations in both copies of the gene to fully disable the brakes. That's the classic 2-hit hypothesis.
2 hits needed.
Although sometimes, losing just one copy called haploinsufficiency can reduce the protein level enough to have some effect. It's complex.
Okay, that framework is really helpful. Proto-oncogenes, tumor suppressors, apoptosis regulators, DNA repair. Shall we dive into those hallmarks now? The first one was self-sufficiency and growth signals. How do they hit the gas pedal?
Right. Cancer cells figure out how to grow without waiting for the normal external go signals. They might make their own growth factors, or more commonly, they develop mutations in the downstream signaling pathways that make them think the signal is always on.
Always on.
Exactly. Oncogenes and the oncoproteins they produce are key here. Receptor tyrosine kinases, for instance, these are receptors on the cell surface that normally bind growth factors. Mutations can make them constantly active, firing off growth signals nonstop.
Even without the growth factor present.
Correct. Or the signaling molecules inside the cell get mutated. A really important player here is MYC. It's a transcription factor, meaning it controls lots of other genes involved in growth, metabolism, making proteins.
So MYC is like a master regulator.
A very powerful one. And it gets dysregulated in many cancers, sometimes through a translocation, like in Burkitt lymphoma, where it gets moved next to a really active gene promoter, or through amplification, where the cell just makes many extra copies of the MYC gene.
Leading to way too much MYC protein.
Exactly. And that pushes cell growth, boosts telomerase activity, which we'll get to, and even promotes stem cell-like properties. We also see dysregulation of cell cycle machinery, like cyclin D and CDK4 complexes, which normally control progression through the cell division cycle. If those are hyperactive, the cell divides uncontrollably.
Okay, so they've got the accelerator floored, but what about the brakes? How do they become insensitive to the stop signals, the second hallmark?
That's where the tumor suppressor genes come in, removing the brakes. The classic example is the RB protein, retinoblastoma protein.
Named after the eye cancer.
Precisely. RB normally acts as a critical gatekeeper, stopping cells from progressing through the cell cycle unless conditions are right. If RB isn't activated, that gate is broken.
And that requires losing both copies, the two hits.
Typically, yes. In familial retinoblastoma, a child inherits one bad copy, so they only need one more hit, a mutation in the other copy in a retinal cell to lose RBF function and develop the tumor. In sporadic cases, both hits happen randomly in the same cell.
Got it. And viruses can mess with RB too.
They can. Certain viral oncoproteins like E7 from high-risk HPV can directly bind to and inactivate RB, effectively disabling that break.
Okay, RB is one major break. What about P53? You hear about that one all the time, the guardian of the genome.
P53 is absolutely crucial. It's a sensor for cellular stress DNA damage, lack of oxygen, other problems. When it senses trouble, it can halt the cell cycle to allow for repairs.
Buys time.
Yes. Or if the damage is too severe, it can trigger senescence, permanent growth arrest, or apoptosis, programmed cell death. It basically tells a badly damaged cell to sacrifice itself for the good of the organism.
So it prevents damaged cells from proliferating?
Exactly. It acts as a transcription factor, controlling a whole network of genes involved in these protective responses. It even influences metabolism. But unfortunately, P53 is the most commonly mutated gene in human cancers.
Most common. Wow.
Inactivated in probably over half of all cancers, either by mutations in the TP53 gene itself, or by other means like overexpression of MDM2, a protein that normally targets P53 for destruction.
So cancer cells find ways to get rid of the guardian.
They have to in many cases to survive and thrive. Life-Rameni syndrome, where people inherit a faulty P53 gene, gives them a very high risk of developing multiple types of cancer, highlighting its importance.
And viruses target P53 too.
Yes, similar to RB. The E6 protein from high-risk HPV, for example, targets P53 for degradation. It's a common viral strategy.
So RB and P53 are the big ones for growth inhibition. Are there others worth mentioning?
Oh, definitely. Many others play critical roles. APC is key in colorectal cancer. Losing it activates the WNT pathway, driving proliferation. TGF-beta signaling is complex. It can inhibit growth early on, but sometimes promotes invasion later.
A double-edged sword.
Somewhat, yes. P10 acts as a brake on the PI3-KKT pathway, another major growth and survival pathway. VHL is involved in sensing oxygen levels and controls HIF-1 alpha. Loss of VHL promotes angiogenesis and the Warburg metabolic shift.
We'll get to Warburg in a sec.
Right. And STK11, or LKB1, is another important metabolic regulator. Mutations cause Peutz-Jeghers syndrome and increase cancer risk. So you see, there are multiple braking systems that cancer cells need to disable.
Okay. Let's talk metabolism now. The Warburg effect, that third hallmark, what exactly is it? It sounds inefficient.
It does seem inefficient at first glance. The Warburg effect, or aerobic glycolysis, is this observation that many cancer cells prefer to metabolize glucose through glycolysis, producing lactate, even when there's plenty of oxygen available.
instead of using the mitochondria for much more ATP.
Exactly. Normally, cells use oxidative phosphorylation in the mitochondria when oxygen is present because it yields much more energy, more ATP. But cancer cells often ramp up glycolysis.
Why? What's the advantage?
The current thinking is that while it produces less ATP per glucose molecule, rapid glycolysis provides the cancer cells with abundant metabolic intermediates, the building blocks they need to synthesize new proteins, lipids, nucleic acids, everything required for making new cells quickly.
So it's about building materials, not just energy.
That seems to be a major driver. It fuels rapid proliferation. And this glucose hunger is actually what PE scans exploit. They use a radioactive glucose analog to visualize tumors because they take up so much glucose.
Clever.
And it's not unique to cancer. Normal rapidly dividing cells also use more glycolysis. Oncoproteins like RAS and MYC actively promote this shift, while tumor suppressors like PTN and P53 tend to inhibit it.
And what are oncometabolites? You mentioned those briefly.
Right. Those are metabolites produced due to mutations in metabolic enzymes like IDH. The classic example is 2-hydroxyglutarate, produced by mutated IDH. This metabolite can actually alter DNA methylation and histone modifications, changing gene expression epigenetically and contributing to cancer.
So the metabolism itself can change the epigenome.
Wow.
What about autophagy in this picture? Cells self-eating.
Autophagy is another complex player. It's a survival mechanism where cells recycle their own components, especially under stress like nutrient deprivation.
So good or bad for cancer?
It can be both. Early on, functional autophagy might act as a tumor suppressor by clearing damaged components. Some autophagy genes are indeed tumor suppressors, but in established tumors, autophagy can help cancer cells survive harsh conditions, like low nutrients or chemotherapy, potentially promoting resistance and dormancy.
So it could help them hide and come back later?
Potentially, yes. So targeting autophagy is tricky. You might want to inhibit it in some contexts, maybe enhance it in others. It's an active area of research.
Okay, so they've rewired their metabolism. Next hallmark. Evading cell death, apoptosis. How do they become resistant to dying?
Apoptosis is a fundamental barrier to cancer. Cancer cells absolutely have to find ways around it. The intrinsic pathway, triggered by internal stress and involving the mitochondria, is a major target.
Mitochondria are key there.
Yes, the release of factors like cytochrome C from mitochondria is a point of no return. This release is controlled by the BCL2 family of proteins. You have pro-apoptotic ones like BX and BEG that punch holes in the mitochondrial membrane, and anti-apoptotic ones like BCL2 and BCLXL that prevent this.
A balance.
A critical balance. Cancer cells often tip this balance by overexpressing the anti-apoptotic proteins like BCL2, making it much harder to trigger mitochondrial permeabilization and death.
So they block the death signal.
Effectively, yes. And losing P53 function is another huge way they evade apoptosis, because P53 is a key activator of the intrinsic pathway in response to damage. Overexpressing MDM2, which degrades P53, achieves the same goal. Even the extrinsic pathway, triggered by external death signals, can be disabled.
And this resistance makes therapy harder.
Hugely. Many cancer therapies, like chemotherapy and radiation, work primarily by inducing apoptosis. If the cancer cells are resistant to apoptosis, the therapy won't be as effective.
Right. Okay, they survive, they grow, they avoid death. What about immortality? The next hallmark, limitless replicative potential. How do they overcome the normal aging clock of cells?
Normal somatic cells can only divide a limited number of times. This is partly due to telomeres, the protective caps on the ends of chromosomes.
They get shorter with each division.
Exactly. Like the plastic tips on shoelaces getting worn down. Eventually, they get critically short, triggering senescence or apoptosis. It's a natural limit on proliferation.
So how do cancer cells bypass this?
The vast majority, maybe 85 to 95%, reactivate an enzyme called telomerase. Telomerase is a specialized reverse transcriptase that can add DNA back onto the telomere ends, maintaining their length.
So they keep rebuilding the caps.
Precisely. This allows them to divide indefinitely, achieving replicative immortality. Most normal adult cells have very low or absent telomerase activity.
What about the other 15%?
They use alternative mechanisms, collectively called ALT, alternative lengthening of telomeres, which involves using DNA recombination to maintain telomere length. It's less common, but achieves the same result. Cancer stem cells, in particular, often express telomerase, contributing to their persistence.
Immortality achieved. But they need supplies. Angiogenesis, getting their own blood supply. That's the next hallmark.
Absolutely essential. A tumor can't grow beyond a tiny size, maybe a millimeter or two, without a blood supply to deliver oxygen and nutrients and remove waste.
So they trick the body into building vessels for them.
They do. They secrete pro-angiogenic factors, the most famous being VEGF, vascular endothelial growth factor. VEGF signals nearby endothelial cells, the cells lining existing blood vessels, telling them to sprout new capillaries towards the tumor.
Creating a dedicated supply line.
Exactly. This neovasculature feeds the growing tumor. And blocking this process seemed like a great therapeutic idea.
Anti-angiogenic drugs, like Avastin.
Yes, bevacizumab, Avastin, is an antibody against VEGF. These drugs can work, slowing tumor growth in some cases, but they haven't been the home run people initially hoped for.
Why not?
Tumors can be clever. They can start using other angiogenic factors besides VEGF or find ways to co-opt existing vessels. Resistance often develops. Plus, the tumor blood vessels are often leaky and abnormal, which can actually hinder the delivery of chemotherapy drugs sometimes.
So angiogenesis is crucial, but targeting it is complex. Now, perhaps the most feared aspect, invasion and metastasis, spreading.
Indeed. This is what makes cancer so lethal in most cases. It's not usually the primary tumor that kills, but the distant metastases. And it's an incredibly complex, multi-step journey for the cancer cell.
The metastatic cascade.
Right. First, the cell has to detach from the primary tumor, invade through the surrounding tissue and basement membrane, get into a blood or lymphatic vessel that's intravasation.
Survive the journey.
Survive in the circulation, which is a hostile environment, then stick to the vessel wall at a distant site, squeeze out extravasation, and then, the hardest part often, establish a new colony in a foreign environment. Colonization.
Sounds incredibly difficult.
It is. The vast majority of cells that enter circulation actually die. Only a tiny fraction succeed in forming a metastasis. Each step presents huge challenges: physical barriers, immune attack, adapting to new surroundings.
Let's break down that first step, invading the local tissue, the ECM. How do they do that?
It involves several things. First, loosening connections to neighboring cells. E-adherin is a key adhesion molecule holding epithelial cells together. Its function is often lost in invasive cancers, sometimes through a process called EMT, epithelial mesenchymal transition.
EMT.
It's like the cells change their identity. They lose their epithelial characteristics and become more mesenchymal, more mobile, more invasive. Transcription factors like sesnail and twist drive this change.
So they become slippery and mobile. Then they need to break through barriers.
Exactly. They have to degrade the extracellular matrix, ECM, and basement membranes. They do this by secreting enzymes, proteases, like matrix metalloproteinases, MMPs, for example, MMP9, others like cathepsins. These chew through the matrix components.
Clearing a path.
Yes. Then they need to attach to the newly remodeled matrix using proteins called integrins and actively migrate. Motility factors, some produced by the tumor cells themselves, some released from the matrix or secreted by nearby stromal cells like fibroblasts, guide this movement. HGF, or scatter factor, is one such factor.
So it's an active process of changing shape, breaking barriers, and moving.
Very active, and often involves crosstalk with stromal cells, the non-cancerous cells in the tumor's neighborhood, which can actually help the cancer cells invade.
Okay, they've broken out locally. How do they get into circulation and travel?
Intravasation, getting into vessels. They can squeeze between endothelial cells or sometimes induce vessel leakiness. Once in the bloodstream, they might travel alone or as clumps, sometimes shielded by platelets.
Platelets can help them.
It's thought they can provide some protection from immune cells and sheer stress. Survival in the bloodstream is tough. Then, at a distant site, they have to adhere to the vessel wall, often via specific adhesion molecules, and extravasate, squeeze back out into the tissue.
And then to grow there, colonize.
Yes, and that's often a major bottleneck. The new environment might not be suitable. Some cells enter dormancy. They just sit there quietly for potentially years before reactivating. We see this in melanoma, breast, prostate cancer sometimes.
Dormancy. That's scary. How did they eventually start growing?
It likely involves complex interactions with the new microenvironment. The tumor cells might secrete factors that educate the local stromal cells to support them, creating a niche. For example, in bone mets, cancer cells can release PTHrP, which causes bone breakdown, releasing growth factors stored in the bone matrix that then feed the cancer cells. A vicious cycle.
An incredibly complex and challenging journey. And all along, they have to avoid the immune system, right? Evasion of immune surveillance, the next hallmark.
Absolutely critical. Our immune system is designed to recognize and eliminate abnormal cells, including cancerous ones. T cells, particularly cytotoxic T lymphocytes, CTLs, are the main killers.
How do they recognize cancer cells?
Through tumor antigens presented on the cancer cell surface. These can be neoantigens, new protein fragments created by the mutations in the cancer cell. The more mutations, often the more neoantigens, which can make the tumor more visible to the immune system. That's why some tumors with high mutational burdens respond better to immunotherapy.
Neoantigens. What other kinds of antigens are there?
Sometimes normal proteins are just way overexpressed in the cancer cell, like tyrosinase in melanoma. Or proteins that are normally only found in the testes or placenta get aberrantly expressed. These are called cancer testis antigens. And in virus-induced cancers, viral proteins themselves act as foreign antigens.
So the immune system can see them. CTLs are the killers.
Primarily, yes. They recognize antigens presented on MHC class I molecules, which are on almost all cells. Antigen-presenting cells, like dendritic cells, are crucial for kickstarting the response. They gobble up tumor material and present antigens on both MHC class I and class II molecules to activate different types of T cells. Costimulatory signals are also needed for full activation.
So if the immune system is geared up to fight cancer, how do the cancer cells escape? What are their evasion tricks?
They have many. One simple way is losing the antigen. If a mutation gets rid of the protein that the T cells recognize, those cells survive. Darwinian selection within the tumor.
Antigen loss. What else?
They can downregulate or lose MHC class I molecules. If there's no MHC to present the antigen, the CTLs can't see the target, like becoming invisible.
Sneaky.
Very. They can also create an immunosuppressive microenvironment by secreting factors like TGF-beta that dampen immune responses, or by recruiting suppressive immune cells like Tregs. And a really big mechanism is expressing inhibitory checkpoint proteins on their surface.
Checkpoint proteins, like PD-L1.
Exactly. PD-L1 on the tumor cell binds to PD-1 on the T cell, delivering an off signal telling the T cell to back off. It's like the tumor puts up a do not disturb sign. CTLA-4 is another important inhibitory checkpoint receptor on T cells.
And that's where immunotherapy comes in, checkpoint inhibitors.
Precisely. Drugs that block PD-1, PD-L1, or CTLA-4 basically release the brakes on the T cells, allowing them to attack the tumor. It's been revolutionary for certain cancers, especially those with lots of neoantigens. Combination therapies are also being explored.
Fascinating interplay. Okay, let's talk about those enabling characteristics you mentioned earlier. First was genomic instability. How does that help cancer?
Genomic instability is like the engine driving the acquisition of all those hallmarks. It means the cancer cell has an increased rate of acquiring mutations and chromosomal abnormalities.
So it speeds up the evolution towards cancer.
Exactly. If the machinery that normally ensures genome integrity is broken, mistakes happen much more often. Loss of p53 function is a major contributor here, since p53 normally halts the cell cycle or induces apoptosis when DNA is damaged.
Right, the guardian is gone.
Defects in specific DNA repair pathways are also key. Think of Lynch syndrome, caused by mutations in mismatch repair genes. These individuals have hugely increased rates of colon and other cancers because simple replication errors aren't fixed properly.
Microsatellite instability.
That's the result, yes. Or mutations in BRCA1 and BRCA2 involved in repairing double-strand DNA breaks through homologous recombination. These lead to increased risk of breast and ovarian cancers because those breaks aren't repaired correctly, leading to more mutations and rearrangements. Even errors by DNA polymerases can contribute. This instability provides the genetic diversity for the tumor to adapt and overcome challenges, like therapy.
So instability fuels the whole process. What about the other enabler, cancer-enabling inflammation? How does inflammation, usually a protective response, help tumors?
It seems paradoxical, but chronic inflammation can create a very fertile ground for cancer. Inflammatory cells recruited to the site release a cocktail of factors. Growth factors, like EGF, that directly stimulate cancer cell proliferation. Reactive oxygen species that can cause more DNA damage, fueling genomic instability. Proteases that degrade the ECM, helping with invasion.
So inflammation helps them grow, mutate, and invade.
And survive. Certain inflammatory cells, like tumor-associated macrophages, TAMs, can suppress anti-tumor immune responses and even protect cancer cells from cell death signals like anoikis, which normally kills detached cells. They can also promote angiogenesis and contribute to chemoresistance. It's like the tumor co-opts the inflammatory response for its own benefit.
Hijacking the system again. Okay, we've talked a lot about mutations within genes. Are there other ways cancer genes get dysregulated? Big chromosomal changes?
Oh, absolutely. Large-scale chromosomal alterations are very common in cancer. Translocations, where pieces of chromosomes break off and swap places, can create fusion genes with oncogenic properties like BCR-ABL and CML, or place a proto-oncogene next to a strong promoter, causing overexpression, like MYC in Burkitt's. Deletions can remove tumor suppressor genes entirely. Insertions can disrupt genes. Aneuploidy, having the wrong number of chromosomes, is almost universal in solid tumors and can alter the dosage of hundreds or thousands of genes.
Aneuploidy.
And then there are more dramatic events like chromothripsis, where a chromosome basically shatters and gets stitched back together randomly, causing massive rearrangements. Gene amplification is also common, making many extra copies of an oncogene like ERBB2-HEO2 in some breast cancers, leading to massive overexpression.
So the whole structure of the genome can be messed up. What about epigenetics? Changes not in the sequence, but in how it's read.
Hugely important. Epigenetic alterations are just as critical as genetic mutations in many cancers. DNA methylation is a key mechanism adding methyl groups to DNA, usually at CpG dinucleotides in promoter regions.
That usually silences genes.
Correct. In cancer, you often see hypermethylation, too much methylation, silencing tumor suppressor genes. Think of it as turning off the brakes epigenetically. Conversely, you can also see global hypomethylation, less methylation overall, which might activate oncogenes or contribute to genomic instability.
So methylation patterns go haywire. What else?
Histone modifications. Histones are the proteins DNA wraps around. Modifying them chemically, adding or removing acetyl groups, methyl groups, etc., changes how tightly the DNA is packed and whether genes are accessible for transcription. Cancer cells often have widespread abnormal histone modification patterns.
Changing chromatin structure.
Exactly. And the enzymes that add, remove, or read these modifications, as well as chromatin remodeling complexes like Swiss-SNF that physically move nucleosomes around, are often mutated in cancer. Non-coding RNAs are another big layer of regulation.
Like microRNAs.
MicroRNAs, miRNAs, are small RNAs that typically bind to messenger RNAs and inhibit their translation or promote their degradation, effectively silencing genes post-transcriptionally. Their levels are often altered in cancer. Some miRNAs that target oncogenes might be downregulated, while others that target tumor suppressors might be upregulated. Long non-coding RNAs, LNC RNAs, are also emerging as major players with diverse regulatory roles, and they're frequently dysregulated in cancer too.
So many layers of control being disrupted. It really reinforces that idea of multi-step carcinogenesis, doesn't it? This accumulation of hits over time.
Absolutely. The classic example is the adenoma carcinoma sequence in the colon. You start with loss of APC, leading to a small adenoma, then maybe a KRAS mutation helps it grow larger, then loss of P53 allows it to become truly malignant in adenocarcinoma. It's a stepwise progression, accumulating genetic and epigenetic changes.
Different mutations happening at different stages.
Generally, yes, although the exact order can vary. We see similar patterns in precursor lesions for other cancers, too. Cervical dysplasia progressing to cancer, for instance. And even within a single tumor, there's often heterogeneity, different subclones with different sets of mutations and epigenetic states, which makes treatment challenging.
Right, contributes to resistance. Okay, let's shift to the external factors, the carcinogenic agents that kickstart or accelerate this process. Chemicals first.
Chemical carcinogens are a major group. Some are direct-acting, they're inherently reactive and can damage DNA right away. This includes some chemotherapy drugs, ironically, which can unfortunately cause secondary cancers down the line.
A known risk.
Yes. But most are indirect-acting procarcinogens. They need to be metabolically activated in the body, usually by cytochrome P450 enzymes in the liver, to become the ultimate carcinogen that actually damages DNA.
What are some examples?
Polycyclic aromatic hydrocarbons, PAHs, are a big one found in tobacco smoke, charred foods, fossil fuel combustion products, aromatic amines and atezo dyes used in some industries, natural products like aflatoxin B1 from moldy peanuts or corn, a potent liver carcinogen, components of betel nuts linked to oral cancer, nitrosamines in preserved meats, vinyl chloride, arsenic, nickel, chromium. The list is long.
So many environmental exposures. What about radiation?
Radiation's another potent carcinogen. Ionizing radiation, X-rays, gamma rays, particles like alpha and beta has enough energy to directly break DNA strands, cause mutations, translocations.
Which from medical scans or radar?
Yes, though the risk from typical diagnostic scans is generally considered very low, cumulative exposure matters. Higher risks are seen in atomic bomb survivors, uranium miners exposed to radon, etc. Different tissues have different sensitivities. Bone marrow, leukemia risk, thyroid, breast, lung are more susceptible than, say, skin or bone.
And UV radiation from the sun.
UV, primarily UVB, is the main cause of most skin cancers, squamous cell, basal cell, and melanoma. It causes specific DNA damage, namely pyrimidine dimers, where adjacent T or C bases get cross-linked. If our repair systems don't fix these dimers correctly, it leads to mutations.
People with repair defects like xeroderma pigmentosum are at much higher risk.
Dramatically higher risk, yes. It highlights the importance of DNA repair in preventing UV-induced cancers.
Okay, chemicals, radiation, now microbes. Viruses and bacteria causing cancer. How does that work?
It's a significant contributor globally. Let's start with RNA viruses. The main example is HTLV1, human T-cell leukemia virus type 1.
Causes leukemia lymphoma.
Yes, adult T-cell leukemia lymphoma, ATLL, but only in a small fraction of infected people and usually after a very long latency, decades. It's endemic in certain parts of the world. The virus integrates into the T-cell genome and has proteins like TACs that mess with cell signaling and gene expression, promoting proliferation. But additional mutations are needed for full-blown cancer.
So the virus starts the process but isn't sufficient on its own. What about DNA viruses?
Several important ones here. HPV, human papillomavirus, is the big one. High-risk types cause virtually all cervical cancers, plus many anal, penile, vulvar, vaginal, and oropharyngeal cancers. They have key oncoproteins, E6 and E7. E6 targets P53 for degradation, getting rid of the guardian. E7 inactivates RB, disabling that key break. E7 also boosts telomerase. Viral integration into the host genome is often involved in progression.
So they directly disable tumor suppressors.
Yeah.
Vaccination helps prevent this.
Hugely effective. HPV vaccination is a major public health success story for cancer prevention.
Great. What other DNA viruses? EBV.
Epstein-Barr virus, very common virus, causes mono, but it's also linked to several cancers. Burkitt lymphoma, especially the endemic form in Africa, where it likely collaborates with chronic malaria infection and the MYC translocation. Also, B-cell lymphomas in immunocompromised people where viral proteins like LMP1 drive proliferation. And nasopharyngeal carcinoma, particularly common in Southeast Asia where EBV is consistently found. LMP1 seems important there too and might make tumors responsive to PD-L1 blockade.
Hepatitis B and C.
Major causes of hepatocellular carcinoma, liver cancer, worldwide. Here, the main mechanism seems to be indirect. The chronic inflammation and liver damage caused by the persistent infection leads to constant hepatocyte regeneration, which increases the chances of mutations accumulating over time. Though viral proteins like HBx from HBV might also play some direct roles.
So chronic inflammation driven by the virus. And H. pylori, the stomach bacterium?
Yes, Helicobacter pylori, a definite carcinogen. Chronic infection strongly increases the risk of gastric adenocarcinoma, again, likely through chronic inflammation. It's also the main cause of gastric malt lymphoma, a B-cell lymphoma in the stomach lining.
Malt lymphoma.
Mucosa-associated lymphoid tissue lymphoma. Interestingly, in early stages, this lymphoma is often dependent on the chronic immune stimulation by H. pylori, and simply eradicating the bacteria with antibiotics can sometimes lead to complete remission of the lymphoma.
Wow. Treat the infection, treat the cancer. Okay, shifting gears a bit. Paraneoplastic syndromes. These are systemic effects, right? Not from the tumor mass itself.
Exactly. Weird symptoms that pop up because of substances the tumor is secreting or because the immune system is reacting strangely to the tumor. They can sometimes be the first sign of cancer.
Like endocrine problems.
Very common. Cushing's syndrome from ectopic ACTH production, often by small cell lung cancer. SIADH, causing low sodium, also linked to small cell lung cancer. Hypercalcemia, high calcium, often from tumors secreting PTHrP, mimics parathyroid hormone. Squamous cell lung cancer, breast, kidney cancers can do this. Hypoglycemia, polycythemia can also occur.
What about other systems?
Neurological syndromes like myasthenia gravis, skin changes like acanthosis nigricans, dark velvety skin patches associated with stomach cancer, or dermatomyositis, muscle inflammation and rash. Bone and joint changes like hypertrophic osteoarthropathy with clubbing of the fingers, often linked to lung cancer. Blood clots Trousseau syndrome, migratory thrombophlebitis, classically with pancreatic cancer, anemia, cachexia.
Cancer cachexia, that's the severe wasting.
Yes. Profound loss of muscle mass and fat, anorexia, weakness, very common in advanced cancers, especially GI, pancreatic, lung. It's driven by inflammatory cytokines and tumor factors, creating a hypercatabolic state. It's a major cause of morbidity and mortality in cancer patients.
Such widespread effects.
Yeah.
So given all this complexity, how do doctors actually diagnose cancer?
It's a multi-pronged approach, but the gold standard remains histologic examination looking at the tissue under a microscope.
Biopsy.
Exactly. Getting a piece of the suspicious tissue via biopsy, or sometimes removing the whole thing, excision. Cytology is also used looking at shed cells, like in a pap smear for cervical cancer, or cells in fluids like urine or pleural fluid. Cytology is great for screening, but lacks the tissue architecture context you get from a biopsy to confirm invasion.
What other tools help the pathologist?
Immunohistochemistry, IHC, is huge, using antibodies to stain for specific proteins in the tissue. This helps identify the cell type. Is it carcinoma, cytokeratin positive, sarcoma, imentin positive, lymphoma, CD markers? It can pinpoint the origin of a metastasis, PSA for prostate, thyroglobulin for thyroid.
And guide treatment?
Absolutely. Staining for hormone receptors, ERPR, in breast cancer predicts response to hormonal therapy. Staining for HR2, ERBB2, predicts response to anti-HR2 drugs. Detecting ALK fusion proteins in lung cancer directs targeted therapy. It's indispensable now. Flow cytometry does something similar but analyzes cells in suspension, great for leukemias and lymphomas.
What about molecular diagnostics, looking at the genes themselves?
Increasingly central, PCR can detect specific gene rearrangements, like T-cell receptor rearrangements to confirm a clonal T-cell population in lymphoma. Evasion fluorescence in situ hybridization can visualize specific translocations, amplifications, or deletions on chromosomes. Microarrays can look at copy number changes across the genome.
And now sequencing, the omics.
Yes, next-generation sequencing, NGS, has revolutionized things. We can do targeted panels looking at hundreds of cancer-related genes, or whole exome sequencing, or even whole genome sequencing.
What does that tell you?
It identifies specific mutations, including actionable mutations that predict response to targeted therapies, EGFR mutations in lung cancer, BRAF mutations in melanoma, etc. It can reveal mutational signatures that give clues about the cause, like UV damage or smoking. RNA sequencing looks at gene expression patterns, which can help classify tumors or predict prognosis, like the Oncotype DX test in breast cancer. We're getting a detailed molecular portrait of each tumor.
It's becoming very personalized. What about tumor markers in the blood?
Tumor markers are biochemical substances, often proteins or hormones measured in blood or other fluids. Things like CEA for colorectal cancer monitoring, AFP for liver cancer, CA-125 for ovarian cancer, PSA for prostate cancer screening and monitoring.
Are they good for initial diagnosis?
Generally, no. Most lack the sensitivity and specificity for reliable screening or initial diagnosis in the general population, though PSA is an exception used for screening. Their main value is often in monitoring response to treatment or detecting recurrence in patients already diagnosed. If the level goes down after surgery, then starts rising again, it suggests the cancer might be back.
And liquid biopsies, detecting tumor DNA in blood.
That's a rapidly developing and incredibly exciting area. Analyzing cell-free DNA, cfDNA, circulating in the bloodstream, we can detect tumor-specific mutations in this cfDNA.
What's that used for?
Currently, mostly for monitoring response to therapy, detecting resistance mutations like EGFR T790M in lung cancer, and potentially detecting recurrence earlier than imaging. The hope is to eventually use it for earlier diagnosis too, but that's still challenging due to the very low amounts of tumor DNA present early on. Analyzing circulating tumor cells, or RNAs and exosomes, are other liquid biopsy approaches being explored.
It really feels like we're entering a new era of diagnosis and treatment.
A golden age, I think. Our understanding is exploding, and it's translating into better diagnostics and more targeted, effective therapies.
This has been an absolutely phenomenal deep dive. Just incredibly thorough. Let's try to maybe quickly recap some of the biggest takeaways.
Well, I think the sheer complexity stands out, doesn't it? Cancer isn't one thing. It's hundreds of diseases at the molecular level. It's this intricate dance between genetics, epigenetics, and the environment.
Right. And the hallmarks framework really helps to understand the common capabilities tumors need to acquire.
It does. And seeing how tumors evade the immune system, that's been a huge area of progress, leading directly to immunotherapy. And the power of molecular diagnostics now. It's transforming how we approach everything.
It really brings you back to that fundamental question about finding a cure. It seems much more about understanding and controlling this incredibly diverse collection of diseases at a molecular level, tailoring treatments.
Exactly. A single magic bullet seems unlikely given the heterogeneity. But the progress in understanding the mechanisms allows for increasingly precise interventions. Control, durable remission, managing cancer as a chronic disease. In some cases, these are becoming more realistic goals. We are definitely making strides.
So looking ahead, what's the next big frontier? Where might the next revolutionary breakthrough come from?
That's the billion-dollar question, isn't it? Maybe even deeper personalization based on multi-omic profiles. Cracking the code of the tumor microenvironment and how to manipulate it therapeutically. Perhaps finding truly universal ways to unleash the immune system beyond current checkpoint inhibitors. Or maybe tackling therapeutic resistance and tumor dormancy more effectively. It's likely going to be advances on multiple fronts.
Definitely leaves you with a lot to think about. If any part of this discussion really caught your interest, maybe the Warburg effect or immune checkpoints or the role of specific viruses, we absolutely encourage you to dig deeper.
Consider this a launch pad. There's always more to learn about this incredibly complex and important field.
Absolutely. Thanks for guiding us through this.
My pleasure.